I'm starting on slide 10 of our PowerPoint in adaptive immunity. When we see this image, what we're looking at here is the process of activation of B cells and T cells. B cells and T cells both are going to originate here in the red bone marrow. And then as you see these arrows and follow along, you see that they move either to the thymus or stay in the red bone marrow. So it says lymphocyte precursors. So these are cells that are formed in hematopoiesis that will eventually become T cells or B cells, will either move to the thymus if they're T cells, or they'll stay in the bone marrow if they're B cells. Think B, bone, T, thymus. Okay? And then it says they will mature there. Maturation is like an education process for B cells and T cells. During their education process, they will go through several tests to determine if they can even detect a specific antigen and if they don't want to attack self-antigens. If they can attack a specific antigen or they have a receptor that will bind to a specific antigen, we say then that B cell or T cell is immunocompetent. If a B cell or T cell doesn't want to attack self-antigens, then we say it's self-tolerant. Once B cells and T cells mature, they will leave the red bone marrow or the thymus and then start circulating in the body, either moving to a lymph node or the spleen or tonsils or somewhere else. They do that, and we call them immunocompetent but naive. They are naive because they have not yet met their antigen that they are specific for. They have not fought any antigens coming in the body yet. And so we say these cells seed secondary lymphoid organs. Primary lymphoid organs, red bone marrow and thymus, are the locations at which these lymphocytes will mature. Secondary lymphoid organs, again, are going to be lymph nodes, tonsils, spleen, that kind of thing. Okay? So as they seed the secondary lymphoid organs and circulate throughout the body, they are doing that to try to enhance the opportunity of meeting their antigen. Okay, so here is a B cell, and let's say this B cell finds its antigen and binds to the antigen. We say that is the antigen encounter. If the B cell here matches up with the antigen at its receptors, then we say that lymphocyte will go through an activation process. As part of the activation, once that happens, the B cell will start cloning itself. That is the proliferation part. So activated lymphocytes proliferate or multiply. And then these lymphocytes start changing into effector cells, which are going to fight the current infection and or memory cells. Okay, so in this case, this is a B cell that finds its antigen, it becomes activated, and then it starts cloning itself, and the clones become plasma cells. So let's go through some of this stuff in a little bit more detail. Okay, so here we see that B cells and T cells, like we said, are both produced from stem cells in hematopoiesis in the red bone marrow. The T cell, cells that develop that are going to be T cells will move to the thymus, and that is where they will mature. B cells will remain in the red bone marrow to mature, but both will then leave their primary lymphoid organs where they mature and start circulating to enhance the likelihood that they will meet a foreign antigen. So in this process of maturation, this education process, these cells will undergo positive and negative selection procedures. The positive selection process in maturation ensures that B cells and T cells are even capable of initiating an immune response. Capability of initiating an immune response would mean that they have the ability to bind to a specific antigen. So they have receptors that could bind an antigen. Negative selection processes ensure that any B cell or T cell that acts against self-antigens meaning that if they bind to an antigen that is a self-antigen, and that is the one that they would want to attack, they're specific for that antigen, we need to get rid of those B cells and T cells. Okay, so after both of these selection processes, if a B cell or a T cell does not pass either one of them, the B cell and T cells will die. Most of this selection process is going to happen even before birth and prenatal development, but it can continue throughout the first year in childhood and actually all the way through life. But it certainly is going to happen more early on in life, even most frequently before birth. So once B cells and T cells leave their primary lymphoid organs and head out into the secondary lymphoid organs or the circulation, then they are going to be ready to be activated. They may never activate, but if they do activate, they first are going to recognize their specific antigen. So what I mean by this, that they may never activate, is if a B cell or T cell is specific for a certain virus that never enters the body, then that B cell and T cell will never activate. But if they do activate, it means that that virus that they are specific for, or that pathogen that they're specific for, is present in the body. So the lymphocytes will recognize their specific antigen in the activation process. And then after that, they will start to clone themselves, like we mentioned before. Every single one of the clones that comes after that recognition phase are going to have the same proteins on their surface that are called antigen receptors. So every clone will be able to detect the same antigen. Here is an example of a T cell receptor antigen receptor on the surface. You'll see here's the plasma membrane of a T cell, and you see there are structures coming out of the T cell that are, it says variable and constant region. This is not an antibody. This is a T cell receptor. But at the tip of the T cell antigen receptor is the antigen binding site. All clones that come from an activated T cell will have this same receptor and have the same ability to bind the antigen binding site. Okay. Notice this blue area here is representing a shape that is specific for a type of antigen. And so the antigen binding site works like a lock and key model where the antigen will bind specifically to the antigen binding site and match the shape. Okay, so this is a T cell antigen receptor. B cell receptors are essentially antibodies on the surface of the cells. And again, they are going to be unique for antigens. So every clone of a particular B cell will have that same receptor on its surface that's an antibody. Okay, so I'm going to stop there for this video, but I will continue with MHC molecules and how we activate B cells and T cells a little bit more in the next video.